Today we're going to be talking about the degradation of proteins. We've made a little model for you guys, and that's us. So this is a protein, and each individual block is a polypeptide. And right there is a exposed degron signal patch. And there are three ways that this can be activated. One, the unmasking of a protein dissociation. Two, creation of a destabilizing N-terminus. And three, phosphorylation by protein kinase. So here we are showing you the unmasking of protein dissociation. So you've got one protein leaving the other, exposing the degron signal patch. Here we've got the creation of a destabilizing N-terminus, and so through hydrolysis you've got part of the original protein leaving so that you've got the exposed protein and degron signal patch. And the final way is phosphorylation by protein kinase. You've got three players: the protein kinase, and a donor such as ATP. So you've got the protein kinase will take a phosphate and attach it to the degron signal and activating that signal. Another player is the ubiquitin ligase, and it's composed of two subunits: the E2 subunit, which is otherwise known as the conjugating enzyme, and the E3 accessory factor that binds to specific signal patches such as degrons. And so these two subunits will bind together and form the ubiquitin ligase. There are three ways to activate the ubiquitin ligase. 1, phosphorylation by protein kinase. 2, allosteric transition caused by ligand binding. And 3, allosteric transition caused by protein subunit addition. The first, phosphorylation by protein kinase, has three players: the E2, E3 complex, protein kinase, and a donor such as ATP. The protein kinase takes a phosphate from ATP and attaches it to the complex, causing a conformational shift. The next way is by allosteric transition caused by ligand binding. You'll notice a ligand appearing at the bottom of the screen. It's going to come and bind with the E3 part of the complex, and it will cause a conformational shift, allowing the protein to come and bind on the E3 subunit. The third way is allosteric transition caused by protein subunit addition. And so this time you'll have a protein appearing at the bottom of the screen that will come and bind with the E3 portion of the E2, E3 complex, and it'll cause a conformational shift. So another player is the E1 ubiquitin activating enzyme, and that is represented with a model right here. And along with the E1 subunit, we also use ATP and the protein ubiquitin. Ubiquitin, using ATP, will attach itself to the cysteine on the E1 subunit, creating pyrophosphate and AMP. The E1 subunit will attach itself to the E2 subunit and transfer the ubiquitin to a cysteine on the E2, and that does not need any energy because it's a thioester transfer. And the protein will come in and it will bind to the E3 subunit, and the ubiquitin will transfer to the lysine on the target protein. The previous process was repeated till you get polyubiquitination, and you'll have two 19S subunits that bind together with a 20S subunit, and the 19S subunit acts as a proteolytic gate. And using ATP, it'll thread the targeted protein through, and the targeted protein will be degraded into amino acids and ubiquitin tags. In addition to protein degradation, the cell uses polyubiquitination for DNA repair, and furthermore it uses monoubiquitination for histone regulation, or it can use multiubiquitination for endocytosis. And you've got a degraded protein.